Hi guys, welcome back to another video. Today we're going to be talking about the cell membrane and the different types of transportation. So, of course, all living cells are surrounded by a cell membrane. Let's look now at the composition and the function of the cell membrane composed of. So it is composed of a phospholipid bilayer, and this phospholipid bilayer has embedded proteins and cholesterol in it. Now, the phospholipids, of course, they're the ones that form the bilayer, and that provides structure and acts as a barrier. Now, proteins also assist in transport, and we will look how different types of proteins assist in transport. We also have other types of proteins that function for communication and also structural support. Now, also our membranes have cholesterol. Now, the reason why we have cholesterol in our membranes is to stabilize the membrane, especially in varying temperatures. Okay, so what is the function of the cell membrane? The function of the cell membrane, it acts as a selectively permeable barrier. What does selectively permeable? It means it does not allow all substances to pass through, so it doesn't allow all substances to pass through. It selects, from the word selectively permeable, it selects what substances should pass and what substances, of course, should leave the cell. Okay, so selectively permeable, as we said, allowing only certain molecules to pass through while preventing others. Now, in order to understand the transportation and why we have different modes of transportation, let's look again at the composition of the cell membrane. So this is a image of the cell membrane. As you guys can see, it's a phospholipid bilayer, okay, and it has two regions. It has the hydrophilic part, okay, so it's has a hydrophilic head, and it also has a hydrophobic part, okay. Now, from the word hydrophobic, okay, phobic means it's scared, so it structures itself in a way to avoid contact with water. So avoid contact with water. That's why you see it's found inside, while hydrophilic does not mind, it's water loving. Okay, so hydrophilic means water loving, and it doesn't mind if it interacts with water. That's why its heads point always to the environment. Okay, now this is important while we look at the transportation of materials. For example, if I have ions that are outside the cell, okay, and I want this ion to pass through the cell membrane. Now, the ion cannot pass through a hydrophobic layer, okay, because we know the hydrophobic layer does not want to come in contact with any polar molecules, okay, or charged molecules too. So we must have a different system, or maybe a protein, for example, that allows this ion to pass through the cell membrane, especially through the hydrophobic part, without having any issues. So now let's look at how do substances pass in and out of the cell. Of course, if I have this is cell membrane, this is a cell with the different organelles, for example. Now I need some materials to enter the cell, and of course I want some wastes to leave the cell. I don't want waste to build up inside the cell. So I have two main types of transport. I have passive transport and I have active transport. Now, from the name passive, what does it mean? It does not require energy, and it moves along a concentration gradient. What does it mean? It moves along a concentration gradient. That means it goes from a high concentration to a low concentration. And some examples are simple diffusion, facilitated diffusion, and osmosis, and we'll be looking at each one of them later on. While active transport, from the word active, that means it requires energy. This energy is usually in the form of ATP, okay. Now, why does it require energy? The reason why it requires energy is because it moves against the concentration gradient. What does it mean, moving against the concentration gradient? That means it moves from a low concentration to a higher concentration. Now, two examples are endocytosis and exocytosis. Now let's look at passive transport. Okay, so as we said, it's a movement of molecules without the use of energy and along a concentration gradient. Now let's first look at the first subtype of passive transport, which is simple diffusion. Now, what happens in simple diffusion? It's usually movement of small, of course, non-polar molecules. Why non-polar molecules? Because of the hydrophobic region we talked about in the cell membrane. Example is oxygen and carbon dioxide. Now in simple diffusion, no proteins, so no transport proteins, let's say it like that, no transport proteins are required. Okay, so for example, let's say this is my phospholipid bilayer, okay, and as I said, which one inside is my hydrophobic layer, and the one pointed toward the environment is my hydrophilic layer. Okay, now oxygen is both a small molecule and a non-polar molecule. Okay, that means it can pass through the hydrophobic region without having any problems. That's why it does not require any proteins, and of course it's going to go from a high concentration to a low concentration. That is simple diffusion. And the same thing for carbon dioxide. Remember, carbon dioxide is a waste product, so carbon dioxide will go from inside the cell to outside the cell also by simple diffusion. Now let's look at facilitated diffusion. Facilitated diffusion, it carries larger or charged molecules to pass through the membrane using proteins. Okay, so why do we need proteins? Because we said large molecules and ions are not the hydrophobic region does not want to interact with them. So we have two types of proteins. Okay, let's talk about facilitated diffusion even more. So facilitated diffusion. Okay, so facilitated diffusion, what are we going to use? We can call it facilitated diffusion or controlled diffusion. So we use proteins, but again, remember, since we're still talking about passive transport, no energy is required, and we go from high concentration to a low concentration. That's why we say down a concentration gradient or along a concentration gradient. Now we have two different types of proteins we use in facilitated diffusion. We have something called carrier proteins, and we also have something called channel proteins. Now what's the difference between both of them? Both of them allow large polar molecules, large hydrophilic molecules, for example, as we said, glucose and amino acids, and also ions, to pass through the phospholipid bilayer. Okay, now we said the two types of proteins are channel proteins and carrier proteins. Now what's the difference between those two? So channel proteins have a fixed shape. Okay, so their shape is not going to change. So for example, I can have, this is my membrane, this is my phospholipid bilayer. Now I left a gap in the middle to show you now. I can have a big channel protein, it has a fixed shape, and this allows hydrophilic molecules to pass through. Why? Because they're not going to interact with the hydrophobic region over here anymore. They're passing through the channel protein. So channel proteins have a fixed shape, and this fixed shape allows them to transport specific charged particles, usually ions. Okay, so usually ions pass through channel proteins. Now what are carrier proteins? Now carrier proteins, they carry their substances. So carry substances, how? By binding to them. So we can have a substance that's outside. Once it binds to the carrier protein, okay, now it allows them to pass through the cell membrane. So the difference is channel proteins have a fixed shape, okay, while carrier proteins do not have a fixed shape, as once they bind to a substance, they alter their shape either to go inside the cell or to go outside the cell. Okay, so they carry substances by binding to them. And finally, we have osmosis. Okay, we're gonna talk about osmosis later on, but know that osmosis is the movement of water molecules, H2O molecules. Okay, so only water moves in osmosis, and also know it's a passive type of transport, meaning no energy is required and it moves down or along a concentration gradient. Now let's talk about diffusion also in general. Diffusion does not necessarily have to occur if we have a membrane. Okay, so if I spray perfume in a room, I can see the particles of the perfume diffuse throughout the room. That's why if I spray at the door, people may be sitting inside the room can smell the perfume. That's because of diffusion. So diffusion is basically the process where molecules move from high concentration to a low concentration. Now, why does diffusion occur? Why is it important? It's important in both animal cells and plant cells. Why? So it's important for gas exchange, and it's also important for nutrient absorption. So for example, why do we breathe? We breathe to bring in oxygen and expel carbon dioxide. Now, how does this process occur? This process occurs through diffusion. So oxygen diffuses into the blood and carbon dioxide diffuses out of the body during respiration. That is an example of diffusion. And also an example in plants for nutrient absorption, water. How do root cells absorb water? By diffusion. Okay, that's why diffusion plays an important role in both animal cells and plant cells. Now we have different factors that can affect diffusion. What are those factors that can affect diffusion? We can have temperature. Now a higher temperature increases the diffusion rate. Why is that? Because higher temperature, what does higher temperature mean? So higher temperature leads to a higher kinetic energy. So higher kinetic energy means the particles are moving faster. So since they're moving faster, they're going to diffuse also faster. So temperature is directly proportional to the diffusion rate. Higher temperature increases the diffusion rate. Now the same thing with pressure. If we have a higher pressure, this will also increase the diffusion rate. Now what about the concentration gradient? Of course, a steeper gradient increases diffusion. What does that mean? So if I have, for example, 10 molecules here, okay, and I have 0 over here, of course there's going to be diffusion. But what if I had 1,000 molecules? Of course the diffusion here, let's call this A and this B, the diffusion in B will be faster because I have a higher concentration on one side, so of course diffusion will be faster. Now what about the distance or the thickness? So of course greater distances to cover slows down diffusion. That's why in gas exchange, okay, so here we said we have gas exchange as a process by diffusion. The cells in our body that are responsible for gas exchange have a very thin membrane. Now why do they have a very thin membrane? To reduce the distance, so to reduce distance traveled by the gases. Okay, so by reducing the distance traveled by the gases, what does that do? That increases the diffusion rate. Now also, if we're going to look at the size of molecules, of course, larger molecules will diffuse slower, while smaller molecules will diffuse faster. Now even the surface area, if I increase the surface area, of course I'm going to increase my diffusion rate. And also it depends on the medium. Now, diffusion usually occurs fastest in gases and slowest where in solids. The reason behind that, it's similar to when we spoke about the temperature. Now, in gases, of course, we know molecules in gases move rapidly due to low density and high kinetic energy. So the particles in a gas are spread out, okay, and they move faster. That's why we have a higher diffusion in gas medium, while liquid, of course, liquid, the particles are going to be a bit closer to each other, okay? They're still moving around, but not as fast as in gases. That's why diffusion will be slower in gases, and that's why it is slowest in solids. Why? Because the particles are tightly packed to each other, okay? They vibrate in place. They're not able to move around like in gases. That's why diffusion is going to be slowest in solids. Now let's look back at osmosis. We said osmosis is the net diffusion, okay? This is very important to know, it's the net diffusion. What does that mean? So even after we reach equilibrium, molecules will still be diffusing in and out. However, the net, so when I sum them both up, I will get zero. It does not mean when I reach equilibrium, no more water molecules are being transported in and out. There's always going to be transport in and out. However, the net result is going to be equal to zero. So osmosis is the net diffusion of water molecules from a high water potential to a low water potential. Now, what does water potential mean? Water potential is basically the measure of the tendency, this should be of water, not if water, of water to diffuse from one area to another. So what does that mean? If I say I have a high water potential, that means the water has a high tendency to diffuse. So if I have a high water potential, means I have a lot of water molecules, so they're going to have a high tendency to diffuse by osmosis to a place where there is lower water potential. Okay, and an example, pure water has the highest water potential. So if I put pure water and I put a concentrated solution, of course water will move from the pure water to the concentrated solution. Now, to talk about solutions, we have three different types of solutions. We have something called isotonic, we have hypotonic, and we also have hypertonic, and we'll be looking at each one of them very soon. But before I talk about isotonic, hypotonic, and hypertonic, let's look at osmotic potential. What is osmotic potential? So osmotic potential is the measure, okay, of the amount of solute dissolved in a solution. That means if I have high solute dissolved, that means I have a high osmotic potential. Now there's an important thing to also note. Now, if I have high solute, that means I have low water. So high osmotic potential means low water potential. So what can I say about water potential and osmotic potential? They are inversely proportional. So now I can add another definition to osmosis. So we said osmosis is what is the diffusion of water, net diffusion of water from a high water potential to a low water potential. Now I can rephrase it and say what osmosis is the net diffusion of water from low osmotic potential to a high osmotic potential. Okay, so this is another definition of osmosis. Now let's talk about the three types of solution. We mentioned isotonic, hypotonic, and hypertonic. Now what is isotonic solution? That means we have an equal solute concentration both inside and outside the cell. So water will move equally in both directions. As you pay attention, water still moves. However, the net movement is zero because it's moving equally both in and out, so the net result is going to be zero. Now what is a hypotonic solution? A hypotonic solution, we have a lower solute concentration outside the cell. So if I have a low solute concentration outside the cell, water will enter the cell. Why? Because water, as we said, water moves from low osmotic potential or low solute concentration. Because remember, osmotic potential is what? Osmotic potential is the measure of the amount of solute dissolved in a solution. So low osmotic potential or low solute concentration to a high solute concentration. That's why in hypotonic solutions we have a low solute concentration outside the cell, so water will enter the cell, which causes swelling. And a hypertonic means higher solute concentration outside the cell. If you guys want to memorize it, hyper, okay, someone who is hyper is always active. Higher, okay, higher solute concentration outside the cell, so water will leave the cell, causing shrinkage. Okay, and now we're going to be looking at the difference between placing an animal cell in an isotonic and a hypotonic and a hypertonic solution versus placing a plant cell. Now, before we talk about that, let's first revise the structure of animal cells versus plant cells. What was one main difference between those two? So remember, we said plant cells have what? They have a cell wall. Okay, this cell wall, remember, is made of cellulose. Now, what does this cell wall provide? We said this cell wall provides rigidity and structure. Okay, and this is very important to know, and we'll see why. While animal cells, we said animal cells, do they have a cell wall? No, they do not. They lack a cell wall. Okay, so now let's look at what happens in an isotonic solution. So we said in an isotonic solution, water moves in and out of the cells equally, so the net result is 0. So if I place an animal cell, nothing is going to happen. The cell will remain the same size as water enters and leaves equally. The same thing if I put a plant cell, it will remain the same because there's no net gain or loss of water. Now this is a question for you guys, and leave a comment down below about your answer. What do you think the medium of our body is? Inside our body, do you think we have an isotonic or a hypotonic or a hypertonic medium? And explain why. Okay, now let's look at hypotonic solution. Remember, we said hypotonic solution, that means we have a low solute outside the cell. So water will enter the cell. Why? Let me show it to you guys. So for example, let's say this is my cell. Okay, I place it in a solution which is hypotonic, means low solute. Low solute means what? Means high water potential. And what did we say? Osmosis, the movement of water molecules from a high water potential to a low water potential. That's why water will move from the solution inside the cell. Now, if I place an animal cell, of course, water is going to move inside, it will swell up, and then later it's going to burst. It's going to lyse, we say, due to the excessive water intake. Why is it going to burst? Because it doesn't have anything to protect it. The cell membrane cannot withstand that much pressure. That's why the cell bursts, animal cells burst in a hypotonic solution. Now what if I place a plant cell? So if we look closely, the cell will become turgid and swollen, but the cell wall, see, a very important note, the cell wall is what prevents it from bursting. Now why is that? Because we have a pushing force, okay? A pushing force against the cell wall we call Turgor pressure, okay? So we have something called Turgor pressure, okay? This presses back, okay? This pressures the cell wall. However, the cell wall is inelastic, so it's going to be able to withstand this high pressure, and that's why it's going to become turgid and it will not burst. Now if I place an animal cell in a hypertonic solution, remember we said hypertonic means high solute concentration, okay? So high solute, we said water will leave the cell causing shrinkage, okay? Let's again look at an example. This is my cell. I place it in a hypertonic solution, okay? Hypertonic means high solute, low water. We said osmosis goes from high water potential to lower water potential. So now I have a higher water potential inside the cell, so water will leave the cell. When excess water leaves, the cell is going to shrink as water leaves, okay, in an animal cell. And also in a plant cell, we say it undergoes plasmolysis, okay, where the cell membrane pulls away from the cell wall because it becomes really dehydrated. So if I want to try to show it to you guys, this is my cell wall, for example, and then my cell membrane, which should be, my cell membrane should be like this, okay? When it shrinks, it's going to look something like this. Okay, you see how it shrinks away from the cell wall. It pulls away from the cell wall. Okay, that's when we place a plant cell in a hypertonic solution. Okay, now let's look at active transport. Active transport is what? Is when we move substances against their concentration gradient. Now, because we move them against their concentration gradient, we need what? We need energy. And this energy comes in the form of ATP. Of course, we also need proteins to help the transport of substances. So they use special proteins and they also use carrier proteins. Now, we spoke about carrier proteins before in facilitated diffusion. However, the only difference here is here we use ATP, while in facilitated diffusion we did not need the use of ATP. Okay, and the reason why we need energy in active transport is because we're moving substances against their concentration gradient. Now, some examples that we can say are important for active transport. Okay, so root hair cells, so in plants. Okay, if they want to absorb minerals, okay, if they want to absorb mineral salts, how do they absorb mineral salts? They absorb that using active transport. So they need energy to absorb mineral salts. Why is that? Because they're moving from a low concentration to a high concentration. So let's write it here. So low concentration to high concentration. Okay, and two examples of endocytosis, active transport is endocytosis. Now, endocytosis, we can split it into two. Okay, we can pinocytosis and phagocytosis. Okay, those are two examples of endocytosis. Now, what happens in endocytosis? It's the process of taking in large molecules by engulfing them in vesicles. So, for example, if I'm going to draw the cell membrane again, okay? If I'm going to draw the cell membrane, it's not the best drawing, okay? And I have large particles, large substances outside. I want to take them inside the cell. So what do? I'm going to start forming like a hole in my cell membrane. So my cell membrane is gonna form like this. Okay, and then it's gonna continue normally. But you see, it's like I formed a hole, okay, where the molecules I want to transport, you see, it's becoming more of a vesicle, it's an engulfment, and then it will complete the vesicle and it will enter the cell, okay? And pinocytosis is usually the uptake of liquid particles, okay, fluid particles, while phagocytosis is performed by phagocytes. Phagocytes are an example of a white blood cell, and they function in fighting pathogens, okay? And of course, this method of endocytosis requires ATP. Now, exocytosis is basically the opposite of endocytosis. So think about it like this: endocytosis, endo, inside; exocytosis, exo, exit. It's the process of expelling materials from the cell through vesicle fusion with the cell membrane. So how does exocytosis happen? I can have a vesicle that has certain molecules I want to expel. It comes in contact with the cell membrane. Once it comes in contact with the cell membrane, it's going to fuse with it. Once it fuses with it, it's going to expel its particles outside. And of course, this process also needs ATP in the form of energy. Why? Because it moves substances against their concentration gradient. Okay, and that's it. If you want, guys, pause the video, try to answer the questions by yourselves, and I will show the answers right now. So make sure to pause the video before checking the answers, try solving them by yourself, and check your answers. Thank you guys for watching the video. I hope you understand everything. If you have any questions, let me know down in the comments below. And don't forget at the end to like, comment, and subscribe the video. Thank you all for watching. See you soon.